Hi, my name is Dr. Jamie Caroon. I am the medical director at the Center for Medical Cannabis Education in San Diego, California. In this video, we're going to look at the first study to show that a high CBD, low THC hemp product administered orally can lead to a positive drug test for THC in healthy adults. This study is entitled Urinary Tetrahydrocannabinol, or THC, after 4 weeks of a full-spectrum high-cannabidiol, or CBD, treatment in an open-label clinical trial. It was published in November of 2020 in the journal JAMA Psychiatry. In terms of background, hemp-derived CBD products are everywhere, and no studies have directly assessed whether the use of these products can yield a positive drug test for THC in urine. It's often assumed that individuals who are using these products will test negative for THC, but the results of this study suggest that that assumption may not be a safe one. This was an offshoot of phase 1 of a 2-phase clinical trial investigating the use of a full-spectrum CBD product in participants with anxiety. Phase 1 is an open-label trial, and Phase 2 is a double-blind trial. The results of these 2 phases have not yet been reported. The open-label phase of the study included 15 participants, and 14 were included in this analysis. The study was conducted at McLean Hospital in Massachusetts between June 2018 and February 2020. Participants self-administered 1 milliliter of the study product under their tongue 3 times per day over the course of 4 weeks for a targeted daily dose of approximately 30 milligrams of CBD and less than 1 milligram of THC. They could not be using cannabis or any other product containing phytocannabinoids. There was no comparison product, and the primary outcome was urinary carboxy-THC concentration. Carboxy-THC is the major metabolite of THC. It is biologically inert, and the cutoff for a positive test was 50 nanograms per milliliter of carboxy-THC. Of the 15 patients enrolled, 14 completed this stage of the trial. 1 discontinued participation because of use of another cannabinoid product. The study population was comprised of 11 women and 3 men, and 12 of the 14 participants were Caucasian. The study product was well tolerated. There were no serious adverse events reported, and none of the participants reported any kind of psychoactivity. Participants were instructed to self-administer 1 milliliter of the study product 3 times a day under their tongue. This translates into about 30 milligrams of CBD and less than 1 milligram of THC. But participants were not perfect in terms of the self-administration, and the investigators used participant diaries and measurements of the weight of the bottles before and after the trial to estimate the actual dose. And so it looks like participants used about 35 milligrams of CBD and less than 1 milligram of THC. And so it was about a 5 milligram per day difference between what they expected and what they actually observed. I published some research on CBD dosage in 2019. I'll include a link to that paper at the end of the video. According to the authors, the study product contained 0.02% THC by weight. They also go on to state that in the US, hemp-derived products can legally contain 0.3% or less THC by weight, which is more than 10 times the amount of THC as the current study product. So to be clear, that 0.3% THC refers to plant material. This is the threshold used by the DEA to determine whether a given cannabis plant is marijuana or hemp. That threshold, the 0.3%, doesn't actually refer to finished products, although many manufacturers try to formulate their products to contain less than 0.3% THC. The regulatory status of CBD is actually quite confusing, and I wrote a paper about this with a prominent cannabis attorney in 2018 I'll also include a link to that paper at the end of the video. I'm on the medical advisory board of CV Sciences, one of the nation's leading hemp-derived CBD product manufacturers. One soft gel capsule of CV Sciences' popular gold formula contains 15 milligrams of CBD, according to the label. And here is a certificate of analysis that corroborates that. It shows that the amount of THC in one soft gel capsule is 0.26 milligrams, and the amount of CBD is 15 milligrams. To compare this to the study drug, we can just multiply by 2.3 in order to convert this 15 milligrams into 35 milligrams, because we know that was the dose that the participants were taking. If we multiply 0.26 by 2.3, we get almost 0.6 milligrams of THC, which is less than the average dose of THC that the participants were taking. Here are the results of the study. 7 of the 14, or 50%, tested positive for carboxy-THC. Participants' carboxy-THC status was only significantly associated with creatinine status. Here's the table from the manuscript. Here are the 7 participants with positive carboxy-THC test results. Interestingly, when the samples were tested again using gas chromatography and mass spectrometry, 5 of the 7 who tested positive for the urine drug screen had levels of carboxy-THC below the level of detection. You'll notice that all of these are below 50 nanograms per milliliter. So then, are these false positives? For example, participant number 4 here tested positive using the urine drug screen, but their concentration was 13 nanograms per milliliter when confirmed by gas chromatography and mass spectrometry. The same is true for participant number 7. Their concentration was 33 nanograms per milliliter when tested again. You would expect that these individuals would test negative given that their levels of THC are below the level of detection for the urinary drug test. The manuscript doesn't mention anything about how the urine samples were stored or processed, and it doesn't report the sensitivity or the specificity of the urine drug screen. But these results, these 5 out of the 7, raise some concern about the possibility of false positives here. And finally, as you look at the bottom of the table, there appears to be no association between a positive test and age, education, BMI, and average dose of the study product. But there is a statistically significant association between THC status and creatinine. So creatinine was statistically significantly associated with a positive test. Creatinine is derived from the metabolism of creatine phosphate in skeletal muscle, as well as from dietary meat intake. It's released into circulation at a relatively constant rate from skeletal muscle, and therefore it's often used as a measure of kidney function. According to a reputable toxicology lab, urinary creatinine determination is an important parameter for evaluating the validity of a urine specimen for drugs of abuse detection. They also go on to say that one of the most important practices used to avoid a positive drug test is the intentional consumption of excess fluid in a relatively short period of time in an effort to flush their systems. Now, these participants have no incentive to try to beat the test in this setting, presumably, but if you look at some of their creatinine concentrations, they're pretty low, which would indicate very dilute urine. 22, 26, 48 is still pretty low, 53, 35. And not only are these values low, but they're also low in individuals with negative tests. And so perhaps these tests are negative because the urine is very dilute. And so maybe there are also some false negative issues here. And with every study, there are limitations. The sample size here is small. There's not a ton of diversity in many socio-demographic variables. The administration of the study product was unsupervised, and the dose was largely based on patient report. and perhaps there was an issue with false positives. With regard to conclusions, it's important for consumers to be cautious when selecting and using hemp-derived CBD products if the consequences of failing a drug test are severe. It's important also to look for certificates of analyses. Many of the companies that are offering hemp-derived CBD products today do not put the milligram amount of CBD on their label in a given serving. And many that do don't actually have certificates of analyses available for consumers to corroborate that, because there have been studies that show that the amount of CBD and THC in these products is different than what is actually listed on the label. So always select a product from a company that is willing to show you their certificate of analysis so that you can compare what it states on the label with what the lab found. Or you could select THC-free products. THC-free products presumably contain no THC, and so the risk of failing a drug test should be zero if, in fact, there is no THC in there. And a certificate of analysis would also verify that. And as always, it's a good idea to consult with a knowledgeable healthcare professional if you have questions or you're confused about how to proceed. With that, I'd like to thank you for your time and your attention. If you have any questions or feedback, please feel free to reach out to me at the email address below.